Cool. So okay, great, perfect. Thank you very, very much for having me. The talks before me were so interesting that I'm a little bit intimidated by adding extra information to this wonderful session. Again, I'd like to thank the organizers for having me today and to give me an opportunity to present my PhD work, which I have been working on at University College London, in London, obviously. The title of my talk is to give you a little bit of a flavor of how we track the single cell heterogeneity in cell populations using deep learning, but also other techniques that we have developed in the lab. The talk, I think, can be applied to stem cells, although the use case that I'm going to be presenting here is going to be a little bit more about cell populations and culture, and about and about cell lines and cell lineages that can be derived from there. So hopefully that triggered your attention. So first of all, the objective of my talk, or the objective of my PhD thesis to start with, was to find out why cell cycle time is different in different cells. So if you ever grown a population of cells in culture, you will actually very soon or very early figure out that there are certain cells which have a capacity to divide very fast. So we refer to them as the fast dividers. And there are certain cells which divide in a much slower scale. So what that means is that the fast dividers in a certain time elapsed have a capacity of populating a large progeny. So they go through multiple rounds of cell division. They go through multiple through very short cell cycle durations, and therefore at the end of the day they populate a larger progeny. Whereas the slow dividers really have their cell cycle time really stretched, and they do not have the capacity to populate as much cells as the fast dividers should or would. So we were really, interested in why such a difference can actually, what the difference originates from. And we wanted to kind of rephrase the question and perhaps ask whether cell cycle duration is heritable in cell populations and in cell lines. So what that means, or to explain what we were trying to study, was to see whether the cell cycle time of an individual cell is dependent or independent from its parents. So from the progeny or from the heritage of that particular cell in earlier generations. So we would expect that if the cell cycle time is dependent in lineages, dependent on the parents, then the cell cycle time would very closely resemble the timing of the parents. Whereas if it was generationally independent or inheritable, it would be completely different, or there would be a distribution of the cell cycle times independent from the parent or from the previous lineage. Well, to be able to actually track cells and do the lineaging across multiple generations, you really do need the tools to actually allow this. And if you want to do this at scale, you actually need a lot of cells to have a look at. One of the options to do this would be to just obtain live cell microscopy and do all of this manually, try to monitor the cells and then draw the lineage trees yourself. But if you really want to do this in a large population of cells across different cell lines and in different experiments, you would probably spend the whole PhD just tracking cells and drawing these lineages. So obviously we try to address this issue, developing computational approaches to this. And the very first step that you have to take when you're actually trying to analyze images in a computational manner is to take those images and take a lot of them. So first of all, we took live cell imaging movies via time-lapse microscopy. We used two of the modalities for our experiments. First of all, we took for our growing cell lines, we took a transmission microscopy, so a bright field image, accompanied by a fluorescence microscopy image, which has a nuclear marker stained with a fluorescent reporter. The cell lines that we're imaging are called MDCK cell lines, which in our experiments should correspond to a healthy, non-cancerous epithelium. And we also have HeLa cells labeled with a fluorescent reporter in their nucleus, which should be representative of a cancerous epithelium or have a cancerous origin to start with. When we set up the experiment, the time-lapse microscopy has been set to take an image every 4 minutes, which actually gives you a very high frame rate. So we take an image of our cells growing in population every cell minutes for a duration of about 80 hours in total, which corresponds to about 3 to 5 days. Those images are usually quite big, which means that they capture more than 1,000 cells per field of view. And we currently have about more than 40 movies per cell line. So this gives us a huge corpus of data and a massive repository of single cell traits and things in single cells that we can have a look at. Now, I've been talking about the computational analysis of our data, so this is what I would like to lead you through. Our automated movie processing pipeline consists of 3 steps at the moment, and obviously, if you've ever done any sort of computational analysis of the images, especially of single cells, the first bottleneck and the first most important step that you have to subject your images through is a segmentation. Depending on what you're looking at, this can be a cell segmentation where you're actually looking at the area and the position of the whole cell, or in our case, because we're using a fluorescent reporter, we actually subject our images to so-called nuclei segmentation. So what that means is that we're translating our input, which is a fluorescent channel or a noisy image to start with, into a binary mask, which tells us where the individual cell nuclei are located, and also tells us how big the areas of the individual nucleus are. This is used to actually identify the cell and the X and Y coordinates, the center of mass, and the nuclear area for every single cell across the duration for the entire movie. If you're wondering about the technology or maybe the method which has been applied to achieve this, we have used deep learning. This step is completely based on deep learning, and the model or the neural network architecture that we use is the famous UNet, which allows you to denoise or segment the fluorescent inputs channel into a binary mask. Later on, we use the binary mask to actually subject these cells to so-called state classification. Now, this may sound a little tricky, and perhaps you don't see the purpose of it just now, but I'll explain why exactly we do this step and why it's, even though it's an optional step, why it's very useful for the later on analysis. In the state classification, what we do is that we take the X and Y coordinates of every single cell which we have segmented in the previous slide. We take it, we crop a tiny patch around that cell, and we subject it. We input these images from both the transmission microscopy and the fluorescence microscopy into a state classifier. The state classifier has the capacity to directly distinguish between the interphase and the mitotic phases, which would correspond to prophase, metaphase, and anaphase. And even though I'm not showing it here, it can also tell whether the cell is dying and is undergoing apoptosis. So what this state classifier is doing is that it's telling you about which immediate phase the cell is in the current patch, in the current field of view. Again, for this step, we have used deep learning, and this model is based purely on a simple CNN architecture. So in this CNN architecture, the input image is translated into one of the 4 labels or 5 labels, which I am showing you here on this slide. Now, I was saying that perhaps this is, you may be asking yourself, okay, why is this needed? And the answer for that question is actually shown right here. So before we actually can connect the single cell observations into lineages, we have to be able to track their positions across movies and also their cell division history. The cell division history means that we are able to connect these cells over generations, and we're able to connect the parents to the subsequent children and the children to their children, and so on across multiple generations. To do this, we have developed a so-called VTRAG algorithm, which is based on 2 steps. It's based on the Kalman filter, which is able to reconstruct the trajectories. So what I mean by that is, as in this schematic, is followed by the magenta line. So what it means is that for every single individual cell in the field of view, this is over time able to connect which cell belongs to the same trajectory. At the end of its cell cycle, at the end of the life of the cell before mitosis, this tracker, the tracking algorithm, is actually also able to assign the cell divisions and to assign the parents to its subsequent children. And the reason why it's able to do that is because we input the cell state classification information into the pipeline. So the tracker is actually smart enough to be able to make these decisions and decide which cell is a parent of which children. Now, we can continue doing this for quite some time, and we can actually continue following these cells over multiple generations. And what this allows us to do is to reconstruct so-called multi-generational single cell lineage trees. I'm showing you a schematic of one on the slide. So here in the center, you have a lineage tree, which consists of one lineage. So you start here at the bottom, at the start of imaging, you have one cell in generation 0, which you start monitoring, and as soon as it divides, you start recording the history and the trajectories of its children as well. You can continue doing this for some time. You can go here. I'm showing you for the tracking for 4 generations, but in our experiments, we have actually managed to follow the tracking and follow the trajectories of generations across more than 8 generations in our populations over the long-term time-lapse microscopy. What is also great about our tracker is that it gives you a frame-wise information, or frame-specific information about every cell at any point of its life. So from the start of the from the time when the cell is born until the time when the cell dies, it can tell you how long the cell spends in anaphase, how long it spends in interphase, prophase, and metaphase as well. Also, it's not just the state of the cell that is important to describe its behavior, but it's also many of the features that you can derive from these cells and from these trajectories, which can be very informative about the cell's life. Just the flavor of them, for example, you may be interested in how the signal is developing over the duration of its life. You may wonder or read some of the behaviors of the cells from the area of the segmented nuclei mask. You may have a look at the local density, how that fluctuates over time, or what actually contributes to the cell cycle time being of that scale. And you can have a look at the displacement, the migrations, the number of neighbors, and all of these features that may actually contribute to the behavior of the particular cell that you're looking at. Now, you may be wondering, okay, well, there are super famous and popular segmentation and tracking algorithms out there. So why is it even worth developing a new one? And here on this slide, I'm not going to go through all of the numbers, and I don't want to really spend too much time about talking about the comparisons. But one of the very important thing that I want to mention here is that we have validated our tracker. We have validated it against very popular tracking algorithms, which have been developed as a general purpose one, such as TrackPy, which is able to track, particles. It doesn't have to be cells. It can be any particle that you want to track over time. And we also benchmarked it against TrackMate, which is a very popular and very surprisingly well-performing algorithm on our data set. But what I wanted to really show you here, or what I wanted to stress out here on the slide, is that we really focused on the multi-generational behavior, or the multi-generational ability to track cells up after their divisions, and we're actually performing quite well on that front. So if you're interested, and if you have a data set that you'd like to monitor over generations, please do consider giving BTrack a try. It's it's an open source algorithm which you can find on GitHub, and you will it's very easily configurable to your own data set. So please do have a look and consider applying BTrack to your own data. Now, if you'd like to hear a little or read a little bit more about the DeepTree, which is how we call the pipeline, and if you'd like to learn a little bit more of the technical details of how the tracking pipeline works, I'd encourage you to go and have a look at our recently, about, I think it's about a couple of days or maybe a week, when this got published in the Frontiers in Computer Science. So please do have a look at our recent paper, and have a look at if this can be applied to your own data, because we try to make it as general purpose as we possibly could for cell tracking. Now, I've been talking about a lot about the tracking. I've been showing you the trees, but perhaps it would be really nice to actually show you what this looks like in practice. So here I'm showing you a movie which should point you to perhaps appreciating that there truly is this intrinsic single cell heterogeneity in the populations. Here, what I did for your convenience was that I colored the individual lineages into 3 colors. So you'll have a blue cell, you have a green cell, and you have an orange cell in this movie. What you're going to be looking at is the segmentation masks of the individual nuclei of the cell in the cell population, and you're going to be watching the movie from its very beginning as we take in from the microscope until the very end, and hopefully you'll see the differences that I was talking about at the beginning of this talk. I've seen this movie many times, and it still kind of puzzles me how good this pipeline is. So hopefully, if this 12-second gave you an overview of the different behaviors of the different cells, and that how the cell cycle time truly influences the division capacity of the individual clones. I'm going to play this 12-second movie once again, just that you can, for example, follow a different cell this time, or you can truly spot the differences. And you can see that the trees, or the lineages, truly result in different behaviors, or in different progeny sizes. So you have an orange cell, which divides only a few times, whereas you have a green cell on the right-hand side, which divides many, many times and actually populates a lot of progeny. Whoops, not again. I'm pretty sure you're bored by that movie already. And so here I'm showing you the single cell heterogeneity, or the lineage trees, which you can reconstruct from watching movies, just like the ones that I've shown you. So here you can start using our tool. You can start truly looking at the differences, at the intrinsic differences in the single cells, in their division time, and you can actually start, correlating, or you can start having a look at the different features which you can extract from these trees, and perhaps lie a little bit of more sense of why the certain cells behave the way that they do. And when you actually look at more than just three of the trees that I have shown you, when you look at as many movies as we have available, you'll actually find out that when you reconstruct these lineage trees, looking at more than 20,000 single cells, you'll actually see this beautiful distribution of the cell cycle durations. And one of the questions that have been puzzling me for quite some time in my PhD is, why is it that certain cells spend as little as 7 hours per their cell cycle, and why is it that some cells actually to take more than 6 times as much to complete one single division? So this is what I'm looking at for the rest of my PhD, but this is probably too much to grasp in one talk. One of the things that I wanted to finish with was that when you have these lineage trees and you follow these lineages for a very long time and for multiple generations, you'll actually figure that you can have a look at the different behaviors and the different relationships within the individual trees. So this is one of the things we've looked at, and obviously the results are in the paper, which we have recently published. But I just wanted to point your attention to the fact that if you look at the ancestral lineages, if you look at the mother, grandmother, and so on and so on, you would expect that if the cell cycle time is truly heritable, and if it follows some sort of pattern over generation, those would be very correlated. Whereas the cells such as the sisters, the first cousin, and the cells that are sharing the generation would not be. However, this is actually quite different. When you look at the ancestral lineages, you'll see that the ancestral lineages are much less correlated than the relationships which are sharing the same generation. So this is just one of the things that I wanted to one of the flavors that I wanted to point to today during my talk. So for the rest of my PhD, I'm going to be studying why that is. I'm going to try see whether the features extractable from these trees can be somehow interpreted as the reasoning why the cells are behaving in a different way, and hopefully we'll manage to see why these trees have such a huge heterogeneity, and why the intragenerational correlations are higher than the generations, and then in the generations from the cell's ancestry. So with this, I'd like to finish up. I'd like to really thank my advisors, Alan Lowe and Guillaume Chagas. I'd like to thank a postdoc in the lab who took most of my data, which I'm playing with on my computer. And I'd like to thank the whole CellX team for their comments and discussions and very helpful suggestions to my project. If you'd like to learn more, please do visit the link of our latest preprint on Frontiers, or either visit my GitHub or Twitter, where I'm very keen on sharing the updates from the lab. Thank you very much.